Hello friends, today we will discuss about the modification of the pre-mRNA into the mature mRNA that is called the splicing. RNA splicing in the molecular biology is a form of RNA processing in which newly made precursor messenger RNA or pre-mRNA transcript is transformed into the mature messenger RNA. During the splicing, introns that are the non-coding regions are removed and exons that are coding regions are joined together. Most of the modification like splicing takes place within the nucleus, either during or immediately after transcription. For eukaryotic genes that contain introns, the introns must be removed before the translation so that the gene can be easily translated into the protein. Look at this diagram. Let's suppose this is the sequence of a gene with 5' UTR and then exon 1 and then intron 1, again exon 1, exon 2, exon 3 and then exon 4, and this is the 3' UTR. We know that the UTRs are the untranslated region, and at the beginning of the gene there is always the 5' untranslated regions, and at the end of the gene there is the 3' UTR, that is untranslated region present. We were discussing about the structure of the gene with the 5' and the UTR sequences and the introns and the exons. Let's suppose this is the exon number 1 presented as E1 and then exon number 2 E2, exon 3 and exon 4. Similarly, this is the intron or intervening sequence, that is I1, and this is the intron sequence number 2, intron number 2, and then an intervening sequence number 3, that is intron 3. So we need to remove these 3 introns from this gene so that translation can occur in a smooth way. So after the splicing, these intronic regions are cut away and the exons are spliced together, and the gene will look like this. All the exons will be joined together. These are all the 4 exons. Exon number 1, 2, 3 and 4. So this is the basic theme of splicing. How the splicing is done. So we know that pre-mRNA is converted into the mature mRNA, and the processing of the pre-mRNA to be converted into the mature form include the 5' addition of a cap and a poly A tail and the splicing. Some genes can be alternatively spliced, leading to the production of different variants of the mRNA from the same niche transcript. So the variety can be achieved by the method that is called the alternative splicing. Splicing is carried out in the series of reactions that are catalyzed with the splicosome. What is the splicosome? Splicosome is a complex of small nuclear ribonucleic proteins that are snRNPs. So these snRNPs and self-splicing clones and ribosomes are capable of catalyzing their own excision from their parent DNA molecules. So snRNPs play the major role in the splicing, and sometimes the ribosome or self-splicing clones can catalyze a reaction that results in the removal of the self-introns. So you can see in this diagram that this is the whole gene, and the introns are cut away that are shown in the blue color. Introns are removed and the exons that are in the black color are joined, and these exons or this mRNA that is in the mature form can be easily converted into the protein. So there is a basic structure of the intron-exon boundary and the structure of the exon. and the intron. We know that always the intron start with the GU and ends with the AG. So this is the splice donor and splice acceptor site. The 5' site always starts with GU, and the 3' side always starts with the AG. The splicing complex recognizes the semi-conserved sequence. We have discussed about the SNRNPs in the previous slides. SNRNPs always uh, recognizes these conserved sequences, and there is always a branch point that contains the adenine. So these three regions are mainly important: GU, AG, and A. And whenever there is a mutation in any of these nucleotides, it results in the false splicing, and sometimes the exons are removed along with the introns, and sometimes the introns are left in the sequence of the DNA. So how snRNAs work? Here, two snRNAs are shown forming partial hybrids. One of these is forming the hybrid at the 5' junction. You know what is the hybrid? Hybrid is the binding of the complementary sequences. So U1 snRNA is binding toward the 5' and the U2 snRNA binds at the branch point A, and this is the AG. We will see in the next slide about the binding of these snRNAs, how they come one by one and then leave. So this is the basic thing how the snRNA binds with the pmRNA. It binds with the complementary base sequencing and it forms a hybrid. So here you can see that this is exon 1 in the yellow color and exon 2 in the green color, and the in the middle there is the intron with this GU sequence, AG sequence, and the branch point. So the U1 protein binds to the GU sequence and the U2 snRNA binds at the branch point. After that, the U4, U6, and UV, the further proteins that are involved in this mechanism, are bound, and the U1 is removed. After the removal of U1, the U2 binds with the U4 and U6. You can see here the binding occurs between these snRNAs, and it results in the formation of the exene of intron and the ligation of exon occurs, and hence this lariat is removed, and when this is removed, it is degraded, and the exons are left only the exon 1 that is in the yellow color and the exon 2 are joined together and the intron is removed. So splicing is the important phenomena that results in the removal of intron. We know that introns do not play any role in the formation of the protein, but they are regulatory regions. They do have some role in the transcription and the binding of the proteins and enhancers, but we have to remove them for the proper translation of the messenger RNA into the proteins. Thank you so much.